There. Hi.
Hi.
Hello. How are you? Here we go. Looks like we've got Harikrishna
Harikrishna. I'm sorry.
and
Harikrishna
Susan.
has a presentation, and so do I. But
Okay,
he has
great.
to leave right away.
Okay, so welcome to the meeting, and we're gonna go to Harikrishna's presentation, then Susan's presentation.
Right. Not exactly a presentation, but just a feature which I wanted to show. That's it.
Okay, yeah, that's great.
Can you hear me, or are you hearing my background noise?
I think it's fine. I don't hear too much of it.
Okay, because anyway.
Okay.
So I accidentally ended up with this feature if I click on a particular part of the cell, it creates like a snapshot of that part zoomed up. So I think that it can be useful if someone want to study two different parts of the cell together, so the snapshots can be used. That's what I wanted to show.
Oh wow.
Yeah.
Yeah.
When number of cells increase, that can be used.
So you can both zoom in and rotate it.
Yeah, that you can rotate it, yeah. But it also gives a snapshot if you record on a particular part. If you click on a particular part.
All right. That's good.
Yeah.
Do you have the coordinates, like when you click on it, or is there a coordinate system available for people if they wanted to know where a coordinate was? Or
Yeah, I can. It works on that methodology. When I click, ray is casted, and it gives the coordinate of the point where it descends, and accordingly the camera shows that point. So I can surely print it at the coordinates.
yeah, I think that would be useful. And then also, like, if people want to do like distances across the sphere, if they find two features, they want to know what the size scale is between them. And the size scale would be the size scale of the embryo, not necessarily the sphere. So,
Yeah.
I mean, we have that information, right, Susan? You're muted.
Yeah, I didn't hear.
Oh. You were muted, Susan.
Sorry, yeah, I'm worried about background noise. Yes, the size of the sphere has been measured, is measured, can be measured. I have a beautiful small ruler under my microscope. So
Yeah.
Okay.
anyway, I could turn off my my picture too here.
Okay.
I don't know if that'll help, but
So that's pretty good, Harikrishna. I'm glad you're making progress on that.
Yeah. Thanks.
Oh, Susan, you
That,
said, oh, was there something
no,
else you wanted to show?
no, actually, I mailed you the proposal which I would be submitting. Like,
Okay. Yeah. Yeah. So if you're interested in submitting a proposal, I recommend you can send it to me a draft. I've gone over
yeah,
in
I've
the.
sent it a draft. I have
Oh,
emailed it.
oh, okay. Yeah. So is that this morning or?
Actually, I think that was morning.
Okay. Yeah. It's probably haven't gotten it yet.
Oh, well, one thing about Hare Krishna's work is that it's going to allow us to do the single cell view of the embryo. In other words, keep one cell at the middle of the picture all the time. And then we can make a network of cell connections and see what kind of dynamics the network has.
Yeah.
okay, what I've got here to present is a network.
Okay.
And it's from the APS physics meeting. So I just copied a few of this person's slides, and I hope I'm not infringing on copyright. I've left everything in place. Should I try sharing my screen?
Yeah, go ahead.
Yeah, okay. Yeah, it's I sent it to you, Bradley, because it never wants to share.
Oh. Well, I don't have my email up, so.
It's on your email.
Yeah.
So sorry. It's oh, there we go.
Okay.
Okay, I got it. Maybe I've got
there
it.
you go.
Can you see it?
Yeah, I can
Yep.
see it.
All right. Then maybe see that?
You're not on the right tab, I don't think.
Not on the right tab.
But the top of the browser, yeah.
Here?
Yeah.
Oh, that's her paper.
Oh.
Isn't as spectacular as her slides.
Oh.
So I'll try the slides again.
Okay. Yeah.
Now, can you see my slides?
Actually, if you click on that, you see on your browser that you're sharing, if you click on that PDF at the bottom, is that the paper or is that the slide? That might be the paper.
This is the paper.
Oh, okay, yeah.
Yeah. And I will try something.
All right.
So I'm going to do this. Now, can you see that?
No, I can't.
Well, darn. If I could say so myself. Like, that's ridiculous. It's not sharing my PowerPoint.
Huh.
Anyways, Nicoletta
So
shared.
you can save the PowerPoint as PDF.
Ah, yeah. Okay, well, I could try that. Okay, desktop, save as, PDF, save, yes. Okay, so can you see that?
Open it up in the browser if you can.
Okay, open it up in the browser. And then we'll go.
There we
Can
go.
you see that?
Yeah, I did. I see it.
Oh,
Yeah.
that's who she is. There she is.
All
And
right.
she took a pipette to some zebrafish embryos and checked their viscosity.
All right.
Ah. Okay.
And then she looked at down the microscope and connected the cells that were together and made these graphs.
Ah.
And then there's the graphs that she made.
Yeah. By the way, we never got it published, but one medical student and I did a thing on zebrafish embryos looking at pigment cells, and they seem to migrate as a loose connection, as the whole sheet.
Yeah, that would be probably mesoderm cells, right?
Maybe.
Would they?
They were black. They were black pigmented cells.
Yeah, pigmented mesoderm.
Yeah.
Anyway,
Yeah.
so percolation is whether something can get in between the cells, and if they're more tightly connected, then they I won't have things
Yeah.
going in between the cells. And oops, I
don't remember whether those zebrafish pigmented cells change neighbors or not. They seem to migrate as a sheet.
yeah, sounds like
Oh, and those were the ones over the yolk, as I recall, not the ones that ended up being stripes. Or
oh, okay. Oh well, anyway, she made a whole bunch of these networks, and the red are pluripotent ectoderm cells, and then the green ones are on the margin are multipotent, she says, mesoderm. So that's that. And then, so the yolk cells are kind of, they're red. And the animal portion are kind of the green here. And anyway, she made these networks and then studied them. And activity and viscosity and a bunch of other things. I didn't show everything because I feel I was trespassing on her copyright. So
Yeah.
anyway, here's the reference, and these are the people she was working with, and I just thought it was fantastic.
Yeah,
Okay,
looks great.
This is an older paper, and anyway, so that's what we have.
Yeah.
Yeah. And what I'm saying is with Harikrishna's work, we could do that for Axolotl also.
Yeah. Yeah, I think so. Well, Susan, I think she got kicked off of the connection. I was going to ask her what year that paper was, but I didn't know. Well, wait till she gets back. Yeah, I think that's great work. It almost looks like finite element modeling in some ways. I don't know if that's what they're doing, like, or that's what they're inspired by, but we had talked about doing that at one time in the group, about doing finite element modeling, which is where you break the surface down into elements and you have these nodes and you have edges, and then I guess you play around with the elements that you get as a result of that process. So you segment it into different pieces and then you play around with it, the physical parameters of those different pieces. There's Susan.
There I am. Yeah.
Yeah.
I give up. It doesn't want to cooperate.
Yeah. So what was...
Good talk.
Yeah, it was. It was a good paper. So what was the year on that paper? I didn't know if...
I think it's 2018.
Okay. Oh, it's not that old then.
No, but her newer work shows these nets that they have developed.
Okay. So how closely related are these nets to finite element modeling?
I don't know. They're I suppose you might be able to use it in that same context.
Yeah. I mean, I was just wondering how they were. Like,
Yeah, each cell could be considered a finite element.
Yeah.
Yeah.
Very nice.
The center of the cell is the vertex of the net.
Yeah.
Yes, but a point is not a finite element.
Right. It's this element that's defined by.
The element's the whole area.
Yeah, well, anyway, the finite element goes from one cell to another. Actually,
One thing, there are papers by Wayne Brodlin where he uses finite elements on an axolotl embryo and claims to determine the forces between cells.
no, she was studying the viscosity of the whole system. And I wish I could show you.
He claims to measure actual forces.
Yeah, well, she was measuring viscosity. That's basically how something responds to force. So how something flows. And
You have those papers, Brett? The ones by Brodlin on finite elements approaches?
I remember seeing it in the citations for the paper that we did, but
Okay.
Yeah,
If you don't have them, send me a note, I'll dig them up.
yeah,
okay, I should look at them as well since I'm doing finite element analysis.
Oh,
Yeah.
okay.
Anyway,
I took the introductory course.
oh, I'm just doing it by trying to make console work. Learn by doing. Anyway, I'll go. I'm sorry that didn't work. Of course not. I sent you the actual PowerPoint, Bradley.
Okay.
In your
Yeah, I'll pull it up later and I can send it out to people if people want to see it. Yeah.
Yeah, it's just a rough view of what she showed. It was way more detailed than that, but copyright, etc. So
Yeah.
okay.
Good. Well, thank you. Yeah, that's great. So, Jiahang or Karan, do you have anything to talk about or ask questions about? I know you're interested in GSOC, interested in have things going on project-wise, or
Yeah, Bradley. I'm still working on the proof of concept part of things, so I thought I'd just finalize that properly before sending the first draft. So I
okay.
think it'll take a couple of more days for me to finalize that part properly.
All right.
Yeah.
Sounds good. All right. That's great. So I wanted to go over a couple again, just kind of repeating what I said last. Okay, Harikrishna has to leave today, so thank you for attending, Harikrishna. That was a nice demo.
Right. Thank you. Bye.
Yeah, bye. I'm going to repeat what I said last week about the projects. So if you're interested in the different GSOC projects, this is GNNs and developmental networks. It's 22.1. This is on NeuroStars. So if you want to join NeuroStars, it's a discourse server that INCF has set up, and they have we have the description here, and we have some sort of conversation about this project. So this one has, people are asking questions. I'm giving them answers here. In this NeuroStars post, or I guess that's what they call it, I have a list of resources here. So these are actually also in the Slack. I've put some resources here for people, some microscopy resources, some resources on GNNs, and somewhere in one of the meetings where this is talked about. So I've also, in the Slack, I've pinned some resources. If you go to the Slack, you go to the DevoWorm channel, you'll see that there's resources posted there or pinned to the channel. So if you look at the pins on the channel, usually it's like there's a little pin icon at the upper right, and you'll be able to find those resources there. The other project is this 22.2, which is Digital Microsphere, and this is the one that we just saw a demo of with the axolotl embryo. So this has, the description, references that you need to become familiar with, and then a couple of, we have the basic data that we've been working with, but we're going to try to get more data later. So this hasn't had as many inquiries, but still. Looks like Harikrishna is working on it, and we might have some other applicants for it as well. The deadline for the proposals is the 17th. So this is something I think it's the 17th. I can't remember exactly what the date is. But it's coming up. That's the point. And so if you want me to look over your proposals, I offer people, if they want to send me a draft of their proposals, they can. So please send it along, and give me a couple days to look it over and give you comments and feedback. Otherwise, that's I think I've gone over in previous lab meetings. I've gone over the outline of what to expect, and it's just a matter of taking your own perspective on it and running with it and seeing where it goes. So we'll talk about that more maybe next week. I don't really have anything more to add, but I just wanted to keep people on top of that.
I have some exciting news, actually.
Okay.
They've developed a small microscope that does optical coherence tomography for the mouth, so you can see oral cavities. And it's a small, slim microscope that looks a lot like the ones that I have in my ball microscope. So maybe one could do OCT with my ball microscope. I just thought I would tell the group that because I'm excited about it.
That's
It's
great.
probably very expensive, but I mean, it's a possibility.
Yeah. Yeah, I imagine it's pretty expensive at this point. The price will come down, so.
Yeah, well, the ball microscope is $1,000 to make, so and I
Yeah.
can't imagine that it would be more like 10,000 to make the other one. So, but maybe you'd only need like two, because you
Maybe.
can see into the tissue at least 1 centimeter or 1 millimeter. Pardon me.
Yeah.
So 1 millimeter depth.
Oh,
Reality. So
that-
maybe just
see
do
inside
two.
of that.
I don't know.
That's great. Okay, so then I wanted to go over a couple of things. The first thing we had was that we Dick Gordon and myself and Jesse Parent submitted I guess we have this accepted as a paper, as a book chapter here. So this is a paper on Basilaria and what we call non-neuronal or aneuronal, or sometimes people call it basal cognition. And so this is the psychophysical world of the motile diatom Basilaria paradoxa. And so the point of this paper was to sort of take, let me go to the actual image of these. So these are our, what was
I can't
that?
see that very well.
You
Sorry.
can't see it very well? If I zoom in,
Yeah,
is it?
make it bigger.
Yeah, there we go. Or is that big enough?
It's readable now.
Okay, yeah, I can maybe zoom in a little bit more. Okay.
Yeah, okay.
So these are so diatoms are very diverse. And I don't know if people haven't seen this presentation before, they're all different types of diatoms. They're algae, they have a silicate cell wall. So they're not like, like C. elegans where they don't, they're animals and they have cells, softer cells. But these diatoms come in all shapes and, variable sizes. They're microscopic organisms, so they're very small. They're on the order of micrometers. If you're familiar with micrometers, which is smaller than millimeters, and you can only see it under a microscope. So diatoms live in aquatic environments. They live in areas near the shore and different locations in the world. And there are different types of diatoms, so it's a very diverse group of organisms. This one is the Basilaria, which is a specific type of diatom. It's this elongated cell type, and they live in these colonies that have this shape, and they kind of, the colonies are stuck together, and they move around. So they have movement, and they can, they can change their shape, or they can move depending on the environmental stimuli that they encounter. So there are things like light. There are things like water chemistry. There are other types of things that happen that this chain of cells or this Basilaria colony responds to. So this paper then talks about, we did a paper earlier on sort of analyzing the movement and looking at what when these colonies move, they move kind of like up and down like an accordion, or they have this kind of rhythmic movement. And we characterized that in a previous paper. But in this paper, we're talking more about maybe what the cells are doing in terms of internal sort of model of what their environment is, how to communicate with other cells in the colony and other things. It's pretty hypothetical, but it's based on a lot of the information from the Basilaria literature, and it's also based on information from some of the neuroscience and neuroscience modeling literature. So we have, it's informed by some of these models that exist for, sort of cognition or what's going on inside the organism. And then we're, going to the diatom literature and seeing what diatoms are doing and how they respond to these things. So most of the theories of diatom movement have really been around physics. And last week I showed this paper on another organism, on a ciliated organism, that behaves collectively based on its physics. So, there's this physical aspect to it where, there are different biophysics going on, and this ciliated organism is responding to it. And, it looks intelligent, but it's really a collective biophysics. In this case, we're proposing that there's maybe this internal world of the Basilaria, that it isn't so much that it's not physics, it's just that there's some information processing that goes on that's happens aside of physics. So this is where this paper goes. We review a little bit about Basilaria and about diatoms here. Talk a little bit about some of these models that we're going to apply. And then we've come up with this model called the collective pattern generator. And I'll get into what that looks like in a little bit. Basilaria in particular and diatoms in general actually have a lot of interesting responses to different environmental stimuli. So these could be like light intensity, water salinity, and temperature. So they have, if the temperature drops, or if I think there are differences in like oxygenation and salinity, there's changes in light concentration because they live in these tidal areas where they get exposure to light in different dosages, throughout the day, throughout the seasons. So they have to respond to that. And this has an effect on their reproduction, which we won't talk about in this paper. But, there are a lot of interesting connections there with what they're, the stimuli that they're experiencing in some of their biology. But that's not really what we're focused on here. But they also a lot of them have to live in these colonies, and they have to sort of communicate with neighboring cells. And that's where it gets interesting, because they do this sort of collective behavior where they're coordinating their behavior with their neighbors. There's a lot of cell-to-cell signaling, which isn't really, I didn't think there was much in the literature on that, but what's interesting is that these extracellular spaces, if you look here at these cells, you can't really see it here, but along the edge there's this extracellular space. And this is where a lot of the interactions are mediated, because there's this sort of adhesive area. So there's an extracellular space that acts as both a site of stigmergy, which is what we might call collective intelligence, and like using different mechanisms in that area to coordinate collective behavior. So stigmergy would be like an ant colony where ants lay down pheromone, and other ants will follow the pheromone, and they'll form trails. They can form this collectively because they have this common signal. So that's what stigmergy is. And these extracellular spaces act to coordinate what different cells are doing when they're interacting or when they're next to one another. And it can also act as a memory medium. And so memory medium can be both like, something like we have in humans where we have long-term memory that has many steps to encode, and you can recall it at any time. Or it can be more like a physical memory, like where you have, something like if you push down on a surface and it recovers to its original shape. That's a shape memory, or they call it shape memory. But it's basically being able to encode that memory in something physical. So there are these mechanisms that sort of exist, and we have to characterize them, and we can maybe link them to some of these models of what we might call basal cognition. So, just to ask the question, maybe, is this some sort of cognition that doesn't exist with the brain? Because these diatoms don't have a brain, obviously. It's just some algal cells that are living in association. So, it doesn't mean that you can't have something that resembles cognition. It's just that they don't have a brain, and we have to be aware of that. They also have these complex behaviors and all these mechanisms that exist. So it may not be simple physics governing this. So this is aneural cognition. In the paper, the term aneural cognition is used. It could be, some people use basal cognition, some people use non-neuronal cognition. It's like the terminology isn't, very tight there, but it's basically the same thing. So there are a lot of papers on this. Binet, who is a psychologist from the 19th century, Verworn, who was another person, I think a biologist from around the turn of the last century. They first proposed a psychophysiological study of protists. So these are single cell organisms, and they're saying basically that they have some sort of psychophysiology. It's kind of interesting that you go way back to the 1800s when people were just kind of working with these organisms. There's not really a strong literature on it, and the first thing they say is, well, there's some sort of cognition here. Okay, so that maybe says that there's something there, but it also may say that maybe people were projecting their own thinking onto the single cell organism. In other words, they're saying that looks like something humans do. So let's say that it's it's basically some sort of little brain inside of the cell. We don't know. So there are two ways to look at that. Either they're onto something, or that they're anthropomorphic projecting. So that means they're projecting human intentions on cell behavior. And so there was some more work done on this. One time, or at that same time, the amoeba was thought to be an evolutionary linchpin that connected the behaviors of single-cell organisms with that of humans. So they said, well, we can use amoeba, which are these single cells, to tell us something about humans. So maybe that was not, the best parallel, but this sort of work continued on throughout the 20th century. And so even today we have work people are doing on stentor, which is a small ciliate, and paramecium, which is another single-cell organism. And so people are working on, these are recent papers that have been published on this. And so people are still looking at this problem. And they're, maybe using some of the mechanisms within the cell to make these claims that, there's some sort of cognition. And so, cognition is actually very broadly defined as information processing. So when they say cognition, they mean that there's some information processing going on. And you say, well, of course there's information processing in the cell. You have genes and you have gene expression and you have all that. They're talking more about more directly related to behavior. And so, there are a lot of ways you can define this. There in cnidarians, you have this, they use a muscular hydrostatic mechanism to link morphogenesis and behavior during the larval stage. This requires tight integration between two biological processes. You also see slime molds where you have a bunch of cells acting collectively. They demonstrate a form of learning called habituation. So in these examples, you see that you can have this sort of behavior both outside of a brain and also in cell collectives where those cells are doing things collectively, but they're also doing things, they're learning, and they're exhibiting learning that looks like something that might happen in the brain. So it's not like that shape memory that I told you about earlier, where you push into a surface and it rebounds. It's more like, it's learning and maybe there's a habituation period. So it exhibits some of these aspects of learning that we see in, say, like there's an organism called the sea slug that people use as a model organism. And the sea slug, you can do different things to its gill slits. You can expose them to water in different ways, and their gill slits have this habituation that they can measure. And it's a very famous biological model for habituation. So, people kind of use that as that's definitely involves the nervous system, and they use that as sort of a benchmark.
Bradley?
Yeah.
That raises an interesting question. First of all, for those listening, Basilaria is worldwide in distribution. You can find just about any body of water. They're especially found in brackish water, where river water goes into the ocean. But we find them locally here, and we don't have an ocean now. So, at any rate, anyone who wants to experiment could try your proposal, for example, with light. We know that if you turn the lights off, they go into the collapsed configuration where all the cells are touching each other, and the shape of the colony is a rectangle or a line of cells. Okay. No one has tried the experiment of changing the period for that. In other words, how often, if you turn the light on and then off and on and off, with a given time for on and off, do they behave the same way, or do they learn to ignore the lights going on and off? Okay, so you can answer questions like what you're asking.
Yeah, I think that'd be an interesting
Yeah.
to see.
There are also questions like, if they're in sediments and they get covered up, do they come up and seek the light? Or if the sediment is drying out, do they go down and seek water?
Yeah.
Okay. Again, this would be very easy to simulate. Even at home, you don't even have a lab for that.
Yeah.
Would it be kind of like photo bleaching if they?
Well, there's a presumption that diatoms do go into the sediment if they find themselves with too much sunlight, to reduce the amount of sunlight. Now, whether Basilaria can do that or not, I don't know. So there's some literature on that. No one has, as far as I know, has done anything with a periodic stimulus like lights going on and off. Okay.
Yeah.
And Margaret Kapinga did that in my lab many years ago, but she only used 12 hours on and 12 hours off.
Yeah.
And so it'll be interesting to see what the response is versus the period that it's on and off. See, if you're a Basilaria living underwater, the light changes continuously. You can't react continuously to short-term changes in light. So it could be that they have some sort of adaptation mechanism to how often light goes on and off.
Yeah.
Okay, so it's an interesting question. You can probe some of these questions you're asking.
Yeah. So yeah, we didn't do that in the paper. We just did like
Oh no one's done it.
Yeah.
Even thought to do it before, but you're raising the question, so I'm just suggesting ways of possibly answering it.
Yeah. So this is this is like an overview of what aneural cognition is, or basal cognition. The reason they call it basal cognition is because the thinking is that cognition, or that information processing, comes from like sort of an evolutionary sort of a precursor. So every organism that has cognition, there's some origin point, or maybe what we see in organisms like diatoms, for example, or single cell organisms, resembles what you'll see later with brains. We know that, for example, that sponges will express synaptic proteins. So those components are there at different parts of the sort of what they call deep phylogeny, or like shared amongst a wide variety of organisms. So we don't really know, and then of course those mechanisms might be configured in different ways the farther you get from, say, vertebrates, where we know what the brain looks like. So, we don't know that. That's why they call it.
A couple of comments there. One is there are some sponges that make light pipes. And so it could be that these light pipes have a function in terms of how the cells respond to light.
Yeah.
Okay. Anyway, you could ask questions like, are Basilaria smarter than people? I'm not sure. Okay, go ahead.
Yeah. Okay, so so there, we have a lot of literature review on this for different things that are, in this area. So we actually have some interesting things on algae and diatoms in this section, where we talk about some of the things that they do. Like Dick was talking about with responding to chemical stimuli, to light. There's a lot more literature out there on this. So there's a lot of literature where people have kind of done basic experiments looking at this, but there's a lot more to do. And like if you frame it in this way, you can ask some really interesting questions, and we don't really know what the response will be. So that's the exciting part of that.
Bradley, have you run across any literature on small or single cell organisms responding to sound or shock waves, or somebody tapping on the microscope or something like that?
Yeah. I have not. I don't think.
We know that single diatoms seem to reverse direction when they hit something. And I'm wondering if they actually feel that they hit something, or are they responding to a bump?
Yeah.
Okay? And,
Yeah.
deliberately bumping them might tell us that.
Yeah.
They had an interesting paper at the APS Physics where they had used sound to create a checkerboard of droplets. And I was thinking of the diatoms, because then they would be trapped in a little droplet, and you could study them. But I'm not sure how well that would work, because the ultrasound drops.
Yeah.
It works for chemical reactions apparently quite well.
Yeah, in C. elegans, the worms actually respond to like vibrations. So people have done experiments with vibrations, and
They have.
yeah. So nematodes don't have ears, but they have mechanosensors, and they have different types of touch sensors. So that's what they're feeling, that vibration.
Yeah, maybe you could send us a paper on that.
Yeah. All right.
I don't think anyone's done that with diatoms.
Probably not. No.
Maybe we can get Thomas Harbich to do it.
Yeah. So there's a large part of this paper devoted to these what we call psychophysical models, and these are like in humans and in other vertebrates. We use psychophysics to investigate the relationship between mental phenomena and physical stimuli. So this is where you have someone who might look at like the intensity of light, and they'll make a response on a joystick, and they'll like select whether they detected that light source or not. And it's just at different intensities. So there's they'll test a range of intensities. You'll respond whether you saw that or not. And then they've been able to create these psychophysical laws that basically show with intensity what human responses to that intensity of light. So you'll usually get some sort of mathematical function that results from this. So that's the idea behind psychophysics. Now, we've introduced a number of different references here to different techniques that we might use to look at psychophysics. So it kind of went all throughout that range of perspective tools. There's a nice review, and I don't have the reference here, but there's a nice review on psychophysics across animals. So they've used psychophysical responses in different animals, including mice, goldfish, and humans. So basically all the vertebrates, and they've been able to work out that it's a cross-species phenomenon. But those organisms all have brains. And so when you don't have a brain, it's a little bit different, maybe a little bit different approach, but you still have the basics. You just look at the different intensities, you look at the stimulus, you look at the response of the organism, and that's basically what you need to do. So we have...
We should call it...
What was that?
Maybe we should call it proof psychophysics.
Yeah. So this goes through a lot of that. Then these different models that we go through, we actually talk about Hebbian learning potentially, and we talk about predictive processing, which is another technique and active inference. So active inference and predictive processing are interesting because what we're doing right now is active inference and predictive processing. We're taking in information, we're processing it, and then we're making a response. And what happens is basically what's going on inside of our brains, or it could be inside of an organism. They have to make predictions about the world based on their sensory inputs. So they're taking in information, they're predicting the next course of action, and then that's what that's something that is adaptive in some way. So this is something that is usually thought of in terms of a brain, but organisms, of course, do this because organisms are anticipatory. They can see, they can respond to things in the environment. And so the question is how would you measure that? And how would you, what kinds of maybe lawful relationships are there? And then if it's not a brain, maybe there's still something going on there with the organism's physiology. But to describe it in this way gives us a framework for looking at that sort of anticipatory behavior. And so I'm going to go down to this last part here, where we talk about a collective pattern generator. So the collective pattern generator is where you build this connectionist network of this organism, of this colony. And you use this connectionist network to model the different points of interaction between the cells. And you have this input, this sensory input coming into the cells. It could be to the front. It could be to any one of these cells, actually. And each of these cells are going to respond independently. If they're in a colony, they're going to respond maybe collectively. But what they're doing is they're basically generating a pattern here in the Basilaria colony where they're going back and forth, or maybe the colony dissolves. But that sort of rhythmic behavior is considered to be what we call a collective pattern generator. So if this lead cell will move up and down, then these cells behind it will move up and down, and they'll have this sort of rhythm where the colony is moving collectively, and that's a collective pattern generator. Now, this took inspiration from what they call central pattern generators. So central pattern generators are things that generate an oscillatory movement. And you see this a lot in insect limbs. So when insect legs are moving up and down, there's a central pattern generator in the notochord that's moving, and it's allowing it to. I shouldn't say notochord; it's a different mechanism, but it's basically a bunch of neurons that are firing together and creating this oscillatory output where the legs are moving up and down, and so they're doing this rhythmically so that they stay coordinated. Your heart also generates a pacemaker. So they're pacemaker neurons that have the same property of a central pattern generation. So it's able to generate your heartbeat. And so this is something that, is, in this case, it's collective. So we're not talking about neurons with formal connections between, formal synaptic connections. We're talking about cells that are behaving collectively. And you could even use this in terms of looking at different organisms. If you wanted to model, say, for example, some social insects behaving collectively. You could use this sort of COPG, which is not talked about in this paper, but you could use the COPG technique to look at those and to kind of look at how those, how rhythmic behaviors in that colony operate. So, and then of course you expect that the COPG will generate lawful responses, things that we can characterize mathematically with respect to maybe some of the fluctuations in the stimulus or some of the external fluctuations that you might find. If you're thinking about a social insect colony, you might have availability of food or, maybe some, stimulus like water or heat or something like that, and it might have an effect. So this is something that is kind of introducing in this paper, but something that might be something to follow up on. And so that's all I wanted to talk about with respect to that paper. I
Bradley,
wanted to
can
go
I
over it.
interject something?
Yeah.
In the 60s, I met the fellow who studied running mechanisms in cockroaches.
Okay.
And he made a startling statement. He said a cockroach has many different ways to coordinate its legs, whereas this modeling seems to settle on one mechanism.
Yeah.
And that might be worth following up. I don't remember his name.
Yeah. Yeah, I'm not sure how. Yeah, I think I remember. I went to a conference. It was like a soft robotics conference, and they had someone who worked on insects, and they used to do these crazy experiments where they'd pull off legs, and they'd see how the insect adapted to it. So they'd pull off some legs, and then and they were able to adapt to it pretty well. So it's interesting how they can do that.
The extreme of that is the octopus, where if you take off one leg, the leg itself seems to behave.
Yeah. Yeah. Okay. All right. Thanks, Jiahang. Thanks for attending. I see he had to leave. And welcome, ABD. Did you have anything to add? Did you have anything to report this week, or?
No, I didn't have anything.
Okay.
I just had some college work, so I was doing that.
Yeah.
So didn't have any progress for anything like GSOC or any other literature study.
Okay. Well, welcome to the meeting. If you have questions about, I mentioned earlier in the meeting that I posted an update in the Slack, so I pinned some information there for people.
Yes, I saw the pinned messages. I'll go through. I didn't got the time because of the colleges.
Okay. Sounds good. Okay. So I guess that's all I had on that. I wanted to go, let's see. Maybe I'll go and talk about, I know there are a lot of papers here we keep talking about, but I wanted to go back to what we had for today. I had a couple of papers that I think Dick sent me, and there were some other things that we've run across in the past here that are kind of fit into a single theme here. And this is morphogenesis, topography, I guess, and complexity. So I'm going to go through these three things, and we'll think about them maybe going on. So we talked about the 1D Izing models, for example, and we talked about morphogenesis and cellular automata. That was several months ago now. And then we also talked about, we've had a long-running theme on patterning in skin and in different surfaces in development. So these papers kind of fit into that area. So the first paper, this is one Dick sent me from John Torday. And John Torday, we did a special issue of biology several years ago, Dick and I. And I remember he was the editor of that. So that was, I remember the name, and then Dick asked me if I knew who it was, and I said, I think so. So that's who it was. So this is morphological forms arising from the evolutionary process are topologies. So this argument here is that you have these morphological forms, and they represent topologies. So what are topologies? And so the abstract reads, the solution to the problem of evolution is proven refractory to experimentation. So how fish have evolved into man phylogenetically, for example, because evolution is thought to be due to random mutations, obviating the opportunity to test such a mechanism, leaving only correlations and associations, which do not show causation. So this is where they're saying, that in evolution, we typically think of things as being due to random mutations, or maybe totally due to random mutations. And there have been people in something called the extended synthesis who have said, well, that's not really the only mechanism for evolutionary change. You have things like developmental changes that mediate some of the mutational changes. You have niche construction, which is actually a feedback between the environment and how organisms modify the environment and evolution. So, we're talking about natural selection here, and we talk about evolution. So what's selecting upon genes? Is it, just random mutations, and then there's some, natural selection on those mutations? Is that all there is, or is there more to it than that? And so that's where the people in the extended synthesis are coming from. So in this case, Torde is talking about some of the morphological features, which is kind of how, maybe we can think about pattern formation. And certainly we have models of pattern formation like the Turing model, the Turing reaction diffusion model, where you have different cells that are, you have these chemical gradients, and the different cells respond to those gradients, and then that drives them to different fates and then creates patterns. So, but you can do like a reaction diffusion model in a chemical environment without any genes at all. You can just see this in a chemical reaction. So, we don't necessarily need genes, perhaps, or maybe there's some interplay there. So, ideally, there would be some comparable process that could be exploited in order to deconvolute evolution, which I guess is this: how do we know what's contributing to it? But none has been forthcoming up until now, having been introduced to knot theory, a subheading of topology. So this is where we're getting into mathematical topology. And so I'm not going to get deeply into that, what that is. It's basically the study of surfaces and shapes. But knot theory is interesting because there are many different ways you can tie a knot. And people have done these studies where they look at knots being made and they try to study the different, it's like a whole fascinating area of mathematics. But topology concerns itself with how geometric objects maintain themselves under continuous deformations. So this is where you have a knot, you have like a string or a rope, and you tie it into a knot. And that knot is just churning that rope in different ways and like making some sort of formation that's stable. So you're basically deforming the rope in different ways. You're folding it over, flipping it, pulling it, and then you have this new shape that is this knot. So it's a complex geometry, but it comes from nothing more than this rope, this straight rope that exists previously. So this is why you might be interested in knot theory and topology when talking about these morphologies. Knot theory is a study of mathematical knots inspired by the knots occurring in everyday life. Now, it occurred to us that since knots tie things together, they are similar in kind to the cell-cell signaling mechanisms that tie physiologic traits together during development, culminating in physiology tied together by homeostasis. So that's an interesting point, that you have these different systems that exist, and they're developing independently, and then they have to be tied together somehow. So you have these different cells that are differentiating, maybe even forming collective. We think about it maybe as sort of collectively forming tissues, but there's also a physiology there that they have to function together in a single organism. And so when you have these different systems that form as they differentiate, we think of this as differentiation, or that they're being pulled apart. But actually, they have to come back together in some way. So now they have to come back together. They have to be physiologically sort of working together, and then they have to exhibit this property of homeostasis, which means that they can maintain stability, which is I think the most challenging thing of all. I think we talked a couple weeks ago about homeostasis and different types of system configuration. But what's really interesting is how these things kind of are put together in development, how they emerge. And so this is where he's going with this paper. So due to competition between prokaryotes and eukaryotes, the latter began forming multicellular organisms through cell-cell communications, mediated by growth factor receptive signaling mechanisms. So it's just that cells are signaling one another with what they call growth factors. Such cellular pathways for growth and differentiation during embryologic development subsequently became the homeostatic basis for physiology. So this is where you get this you get these mechanisms that are going on at the cell level, and they're building to this homeostatic, these homeostatic mechanisms. In the process of doing so, the cells involved produce extracellular matrices that stabilize them physically, forming true knots that literally tie together these cells as organisms, or these systems as organisms. So during the process of chronic disease, such cellular communications break down, reverting back to earlier developmental and phylogenetic homeostatic stages of evolution. So basically you get this homeostatic process that emerges through a lot of this cell-cell communication signaling. You get growth factor exchange between cells, between tissues, and this sets up this sort of relationship for homeostasis. And there are a number of people who have argued that one of the things that's really important in health is maintaining homeostasis, or the ability to maintain healthy tissues, healthy bodies, is this sort of homeostatic ability. And so, it's kind of magical, because it's like, how do you the body, for example, maintain, humans and other homeotherms maintain their body temperature, but it isn't like a given that that's the case. I mean, that has to happen through a number of complex mechanisms. Those can break down in some cases. It's with environmental stimuli or with some other dysfunction. So, this is something that is maintained in development, continues through adulthood, and we have to understand kind of how that worked, or else, complex organisms, they had to evolve this ability as they evolved, or else they couldn't really maintain, we couldn't maintain very large bodies without some of these homeostatic mechanisms. So this is one example here of formation of micelles from lipids in water. So this shows like you have a simple system of a micelle that is like a empty cell. It's basically a lipid bag, and you can end up with a chemical homeostasis here as it's pushed down into the water away from the atmosphere. So you can get these, they have these things called minimal cells, which are an experimental technique that people use to look at like how cells maybe evolved.
Bradley?
Yeah.
There's one small area you made there. A micelle is not a vesicle. A micelle consists of the charged molecules on the outside, and the hydrophobic parts are inside, but the micelle has essentially no water inside.
Okay.
Whereas a vesicle is a layer of amphiphiles with water outside and inside.
Okay.
And I'm surprised he didn't show both.
Oh, okay. Anyways, yeah, so this is sort of, this is just a diagram of this process.
My cell's more like a hairy ball.
Oh, okay, yeah.
Okay.
Then it gives an example of endosymbiogenesis, which is where, over the course of evolution, cells have internalized factors in their environment that pose existential threats. So these are X, Y, and Z, and these factors become compartmentalized over time. So you get this compartmentalization over evolution, where things become incorporated into the organism as you go through evolution. And so it allows for organisms to fend off threats more effectively, as opposed to if all these, so like, you might have like X, Y, and Z as separate organisms living in association. If they live in the same organism, they can fend off threats more easily. So this is one of the compartmental, and the same thing happens in the organism. That, if you have things in the organism that are not working together, having them work together is a better strategy for fending off external threats. So if X, Y, and Z were all systems that interacted in different ways, they form like a circuit or something else, this is something that would help the organism. And so finally, this relationship between topologic knots and cellular evolution. On the left is a schematic for knots as simple circles that can then form complex configuration, but then can be unknotted to reform as circles. So you can have these knots that can reconstitute back into cells. So this is the idea of knots. I don't really know if this knot is supposed to be like, it's basically integration of different things. So he's showing that it's reversible in some cases. So this is interesting, goes through a lot of things here in terms of the sort of the emergence of these different things. So the evolution of heart, lung, and kidney. You have the lung here that shows, it evolves in different ways in different organisms, but you have these basic components. You have the heart and the kidney. So, and note the increasing complexity of these phenotypic traits from left to right due to the interaction of a changing environment and the cellular components of each structure. So, now the next two papers I'm going to go over a little bit more quickly because we don't have a lot of time left. This is lizard skin patterns in the Izing model, and so we talked about this several months ago with Tom Porteges, and this is this 1D Izing model that we talked about, and this is a paper on lizard skin patterns. So this is where you have lizard skin has a lot of nice patterns on it, and they form through this sort of morphogenetic process. So in this case, the ocellated lizard exhibits an intricate skin color pattern made of monochromatic black and green skin scales. The dynamics of color flipping are known to be well modeled by a stochastic cellular automaton. So there's this intricate pattern of scales. They flip their colors based on some process, the stochastic process, which is basically as simple as flipping a coin. Flipping a coin is a stochastic process, and it's a random process, but it's that process of generating that random state, which is the idea behind stochasticity. So people talk about stochasticity all the time in biology, but you can apply it to a lot of problems. But here we're applying it to a cellular automaton where you're making a decision between what color a scale should be, and then this is happening in parallel, so that the scales have different patterns. We show that the wait time probability distribution of the pattern corresponds to the canonical probability distribution of the Izing model and can be generated by dynamics different from the commonly used Glauber model. We comment on the skin scale patterns generated by the Izing model on the triangular lattice and the low temperature limit. So they talk about, let me see if they have any images here. So this is an image of scales here on this organism. And so this is the dorsal surface, which is the back. So you can see the top of the organism or the back. This is what it looks like. So you have this patterning that goes from 29 weeks to 162 weeks. Basically, you have this almost looks like multicolored corn, if you've ever seen that, with different scales, different colors, and then you see that this changes to like a bicolor pattern, and then you get the spacing in between the pattern. So it's like
Bradley,
yeah.
Stupid question. How they guarantee on those magnification pictures at different weeks that they're looking at the same part of the lizard?
I don't know. It doesn't really say. Yeah.
Okay. I mean, they look similar, but that's not a proof that they're exactly the same portion of the lizard.
Right. So I don't know. Yeah, I don't know what. Yeah. Because you have to get like right on that spot, exactly in that spot each time, which is a little hard to do. I mean, they show the box, but it's like
unless they have intermediate steps and they're only showing these.
Oh, maybe, yeah, that could be.
Yeah.
So they wanted to compare this pattern with something that they would generate in a cellular automata. And so this is where you have the simulation, this green function, which is the number of neighbors of same color. So if you look around each of these scales, they have neighbors. And the neighbors should be like either the same color or a different color. And so the idea is that if it's the same color, it's more homogeneous. If it's a different color, the model is more heterogeneous. And the rules of the system may or may not tell it in the next step to become more like its neighbor. That's the idea, is that, this sort of morphogenetic process, you want to compare it to a cellular automaton. And so the question is whether it behaves like that. And it actually behaves.
Another stupid question. A single point there of a given color is a scale?
I think so, yeah. Yeah, so these are all scales.
And how many cells are there in a scale?
I don't know if they tell us that here.
And the other question, instead of the cells flipping, could it be that they shed the scales?
Yeah, they could, yeah.
And then they have to grow new ones, and the new one maybe is where the flipping occurs.
Yeah.
I have no idea if lizards shed or not, except for the whole fin.
Oh yeah, they shed their scales.
I know snakes better.
Yeah,
skin.
I think so.
Lizard.
But I don't know what the time scale. I don't know what how often they do it. Right?
I'm just saying, you got to be careful interpreting what these dots mean.
Right. So yeah, they don't really say how many cells constitute a scale.
And whether the scales are continuous or they get lost and they have to be regenerated.
Right. So yeah, and the comparison they make is the simulation versus the observed. So the simulation, the number of neighbors of same color, the frequency is higher earlier, and in the biological example, it takes a little bit longer for it to reach its maximum frequency. So the frequency would be like, if you have a bunch of neighbors, how many of those are of the same color? If it's 0.2, then that means on average it's like maybe the 2 out of 10 or 1 out of 5 neighbors is the same color, and so that's where the frequency comes in.
Yeah.
So
the other thing is are the black dots scales or of a different color, or are they something else?
I think they might be scales of a different color. I don't know why they change, because they don't have very many black scales in the 29 weeks. But yeah,
lots at 162, and the light colored scales are gone by 162. Well, by 61 weeks.
So it says flipping is a function of the number of green and black nearest neighbors, which are the scales, green scales and black scales. Yeah, as a function of the neighborhood. So this is the green scale and black scale flipping probabilities. So you can see they go up with these parameter values in the simulation, and then they look at the biology to try to match it.
They look hexagonal cell, like hexagonal shapes, until the last picture that doesn't show that very well.
Oh, this is human here? Yeah, you don't see the shape as much.
Yeah.
Yes. Yeah. Let me see if they have any other images that might be informative. Well, this is just where they show the simulation here. So this is where they show the hexagons, but this is the stochastic cellular automata, and then they show the single flip Izing actually is another model for this. So
they don't include the light green scales.
No.
In this
No, just the green and the black in this one.
Yeah, because it's obviously a 3-state system, at least.
Right. And I'm not really sure whether they just focused on the 2 states. Maybe it's easier, or maybe it's like more relevant to later development. But
well, the initial Izing model was a 2-state model.
Yeah.
It may be a bias.
Yeah. So let's see if there are any other images here. So this is the parameter space of the triangular Izing model in polar coordinates. So this is where they kind of go through some of these. I'm not sure what these polar coordinates are, but like you have the black state, pure green state, and then you have like a black where black dominates, and then green dominates, and then you have the shift, in the density of the cells from one to the other. So you can see that like the green dominates and then black starts to dominate when the black cell or the black scales become more prevalent. And so they place this out on a radio map so that they can show some of these. This is something I think with respect to the Izing model and the temperature. The Izing model being a rough approximation of a physical system. So that's why they use that kind of an analysis. So the red star indicates the location of the optimized lizard Izing model at finite temperature. So you can see here that this is where the optimal model is. So this is a very challenging paper, I can tell. It raises a lot of questions, but maybe someone...
It has Gibbs energy in it too.
Yeah.
Yeah, it looks like it's a transition from light green to dark green. In 48 weeks to 61 weeks.
Yeah.
Okay. Is there any transition from light green to black?
Yeah.
I don't know. You'd have to check it dot by dot.
Yeah. So this is something we can follow. Maybe someone can follow up on it. I can send the papers out and we can look at it more. I don't know. Maybe Tom has something to say about it too, since he's done work. The final paper, Epigenetic Forest and Flower Morphogenesis. So this is a paper on flower morphogenesis. We don't talk much about plants in this group, but this paper studies the epigenetic process that leads to angiosperms flower architecture, or flowering plants. So these are flowering plants. They have flowers. It's a specific type of morphology. It has like a lot of these patterns, like you see with a lizard scales. In this, as a case study, we analyzed the flower of Arabidopsis thaliana. They look at the gene regulatory network obtained during sulfate determination in the early stages of the flower's development. So they constructed this gene regulatory network from a previous work using experimental data. They start by constructing and analyzing what they call an epigenetic forest, or a discrete representation of Waddington's epigenetic landscape. And this is obtained as the transition graph of a discrete dynamical system that they associate with this genetic regulatory network. So the GRN, or the genetic regulatory network, is this system where it's producing out dynamical outputs, and they can use what they call a DDE, or a discrete dynamical set of equations, to model that. Next, we propose an optimization problem to model morphogenesis by defining a biologically meaningful function that accounts for the work involved in cell specialization. Finally, they solve this using a genetic algorithm. So they simulate it using a genetic algorithm. The optimal solution found by the algorithm correctly recovers the flower's architecture, as observed in wild-type flowers, and recovered in other theoretical work. And then the method is directly applicable to the other GRNs of the attractors, which are mathematical constructs, consisting of equilibrium points only, and could be extended to the situation where there are periodic attractors. So they're looking at morphogenesis in this way through like this genetic regulatory network that's producing this output. They're analyzing using this differential equation, and then they're using this model of Waddington's epigenetic landscape, which is, then how they analyze this. So the different states that can result. So this is a picture of the flowering. This is an angiosperm, the typical anatomy here. And then this is a directed graph that represents a Boolean network. This is the gene regulatory network that they use. And we've talked about like different ways you can do this. This is just the way they've managed to do this. This is from experimental work. So there are a lot of interactions here between different genes. And the question is like, between these interactions, what are the outputs that we can model? And so they use this approach called epigenetic forests, which is based on the epigenetic landscape, which is this landscape where you have the ball and it falls down into these different wells, and that's the trajectory of the phenotype. So we've talked about this before, but it's basically, it's similar to some sort of like energy landscape, or fitness landscape where you have these paths, and there's actually a tree underlying this because the idea is that you want to characterize different branch points based on these the output of these GRNs. That's kind of the idea. So you have these branch points in the landscape that lead you down different paths. Here's an example of the epigenetic landscape, and you can see these branch points where the ball either goes one way or another, and it goes down this path, set of paths at the bottom here where you have like 4 distinct paths. So that's what they're doing here. And so the discrete dynamical system is shown here. They don't show the math, but they show like that as your state increases, your state changes from I to I plus 1, the state of the matrix changes here, and you get this, you model these transitions as a discrete dynamical system. And then you can build a transition graph where you can show transitions between states. So then they have these different equilibrium points for different things, for sepals, petals, inflorescence, stamens, and this kind of shows you what these states are. I think if different genes are on or off, they produce these things. So I'm not really sure, but this is what they call a genetic encoding. So, you're going to encode your morphology into this genetic regulatory network, and then you're going to simulate the genetic into a genomic encoding, and then you're going to simulate the genetic regulatory network, and then you're going to plug that into a differential equation. That's sort of the order of operations on that. So the tree corresponding to sepals, it looks like this. You get this, you get this pattern of sort of it's converging down to a certain pathway here. You can see it does this recursively. And then you have petals. So this is the pattern for petals. And again, this is the state transition matrix here, the state transition graph. This is just turning different genes on and off for these different structures. And then they use some other things. I'm going to see if there are any other nice images here. So this is the epigenetic landscape where you have 4 schematic trees depicted on Waddington's landscape. So this is where you have these trees that sort of define these endpoint nodes. They just show like the node, the root of the tree, the chain node, and the chain edge. So the orange path together with the yellow nodes is a path. A chain and red nodes correspond to the stable states. So these red points are the stable states, and they built upon these different dependencies coming down. And then this orange path is sort of puts these things together, I guess. So it's another really dense paper, but I can send this out to people. You can take a look at it if you're interested. You can follow up on it later. So that was a lot, but I wanted to go over those. I thought those were some interesting papers.
Everyone should have attended the APS physics meeting, though. In the biophysics, it's about what you talk about every week. It's amazing. It was amazing, and I haven't even shown you even a drop of it yet.
Oh, yeah.
So reference, and these are the people she was working with, and I just thought it was fantastic.
Yeah, that's very good. Yeah.
Okay, can you put that reference in the email?
Or I can put it in the chat too here. I'll get it back up. There.
Copy. Free.
There. Okay, I just had to have the PDF open before I tried to share screen. So now I have a method of doing this. I'm so pleased. Finally, able to share screen.
Great.
Okay.
All right. Yeah. Well, thanks for that. That's great. Okay. Well, thank you for attending the meeting. I'm going to send out some emails and see you next week. Thanks.
Yes,
Okay.
thank you.
All right, see you. Bye.
Great session.